Welcome, everybody.
Yeah.
I am delighted and honored to introduce Doug Lauffenburger. I'm going to start with a very short summary of some of his major awards and recognition, and then move on to more personal things. So Doug started off summa cum laude at University of Illinois. He's a member of the National Academy of Engineering, the American Academy of Arts and Sciences, the Biomedical Engineering Society. He's also a member of the American Association for the Advancement in Science, and the list goes on and on. He is a founding father of the Department of Biological Engineering. He's also in the Department of Chemical Engineering, Biology. He's a member of the Koch, the Raygon, the Center for Gynepathology, the Center for Biomedical Engineering, which he helped to found, and the Superfund Research Program. So I originally invited Doug to speak at the Superfund Research Program meeting, and then I realized he really deserves a much bigger audience. And so this presentation is sponsored by the Department of Biological Engineering, Chemical Engineering, the Koch, the Department of Biology, and the Superfund Research Program. So, nearly 30 years ago, Doug had a vision. He wanted to use engineering principles and mathematics to study and understand biology in new ways. At that time, most of the people in the life sciences were studying one protein at a time or one gene at a time, and people weren't convinced that you could get meaningful information from large data sets because they didn't think you could get results if you didn't have hypothesis-driven research. And Doug saw that with huge data sets, you could not only get meaningful data, but you could generate new hypotheses that really move the field forward. So Doug set out to change the way people think about data, and ultimately he helped people to think about other kinds of big data sets and how they could all be integrated. So Doug's original vision was to bring together biologists and engineers, and it was really way, way, way ahead of its time. And at that time, we kind of felt like we spoke different languages. And I didn't think I had very much in common with someone in engineering and vice versa. But Doug had a vision to bring us together in a new way. And he was sure that if we kept working on it, he would help to bridge the chasm between biologists and engineers. And he envisioned that we would eventually start working together. So Doug joined with Steve Tannenbaum. Steve was the director of the Division of Toxicology. Toxicology is very solutions-oriented, and it's very cross-disciplinary. And at that time, people were sort of siloed. They were chemists, or they were biologists, or they were biochemists, or physiologists. But toxicologists span the whole range, from atom to animal to population, and really focus on real-world problems and solutions. And Doug realized that he not only needed engineers, he needed biologists, and he needed people who are thinking about the bigger picture and trying to take on some of life's major challenges. So we started with a handful of graduate students and no major. The vision that Doug had was that biological engineering could ultimately become its own discipline. And together with Linda Griffith's remarkable insights and profound dedication, a curriculum was developed for the first time in this burgeoning field. Linda won the MacArthur Genius Grant for her visionary work, especially in curriculum development. Doug was not only a visionary, he was truly a great leader and is a great leader. And if I went to Doug and I had some issue or needed something, he almost always honored it if it was a reasonable request. But he really lifted us up. He believed in us. He championed us. He was compassionate and supportive and helped us to see our own potential in ways that we couldn't see ourselves. Doug really, really was a great leader and continues to inspire many, many people. So amazingly, Doug's big visions have mostly come to reality. The chasm between engineers and biologists has indeed been bridged, and BE not only became the first major in over 20 years, but it became a guiding light for dozens of other programs around the country and even around the world. So I remember when this room was mostly empty because we were a very small group, but Doug was right. We did come together. We did create a new major, and we have truly fused biology and engineering. Finally, we're here not just because of Doug's vision, but because he was tireless in his dedication, and he was the most distinguished leader we could possibly have had in those early years, and he is continuing to be an incredible inspiration to many around here and around the world. So with that, thank you so much, Doug.
Thank you. Thanks, so appreciate it. Is this off now? Yeah. Thank you, Bevin. Really appreciate the very kind introduction, and especially like to return the admiration to you for your really extraordinary contributions to our community in so many ways, so really admire you tremendously and appreciate you as a colleague and friend. And speaking of colleagues and friends, it's fun to look around the room. Yes, I do remember back when there was hardly anybody here for anything. You'll see something like that along the way. But a couple nice sectors of people. I see some colleagues who were there at the founding. Really appreciate you coming back for this. And you can correct me if I've got anything wrong. So feel free to do that. But it's also fun to see new students who weren't even born yet when we this is not only history, it's prehistory in a lot of ways. So it's really gratifying to have the chance to explain this history because, we lived it, and for us it was our day-to-day challenge. And for you all, it's like, what's the big deal? There's a department. Cool. So I'm happy to have this chance. I hope it'll be interesting to you, and I'm very appreciative that so many folks showed up. All right, so what are we going to do? This is a guided tour, and it is a history of how BE, MIT Biological Engineering, came to be. In terms of the epoch of history, it'll be about 1975 to about 2005. Okay, and there's reasons for that. Once we hit 2005, we felt good about what we had here. 1975 was about when ideas were occurring to me, so that's the reason for the selection of that period. All right, so what is the big deal? What are we here for? And it really is this. Something very radically new happened at MIT. Starting in about 1998, that had to do with the landscape between biology and medicine and engineering. And we have established a quite unique biology-based engineering discipline that we call biological engineering. As you'll see, it's grounded in the mechanisms of biology, the molecular cellular level. Yet the way of thinking about it is the way engineers think about things. You've got to quantify, you've got to predict, you've got to embrace complexity. You're not trying to simplify things and throw them out. You're trying to say, no, we want as accurate a predictive capability for what we design. And so what we're after is creating biological technologies. And there have been new degree programs established, and most singularly the new undergraduate major that some of you are in, and it started in 2005 and was the first new major in almost 4 decades, actually. So whether you recognize it or not, that was a big deal, and as Bevin said, it was brand new on the scene in engineering, really a new approach to moving biology forward, and it really has been quite influential. All right, yeah, but where did it come from? What had been in this landscape before? Well, biomedical engineering has been around for a long time. The 50s for sure, when artificial organs, artificial kidneys, artificial hearts, artificial lung, oxygenators, prosthetic implants. Then as we moved into the 60s and 70s, robotic surgery and drug delivery, there were many biomedical engineering departments around the country. This was a well-established field. I want to use the word carefully. But what it was, if you think about it, it was classical engineering. This is engineering where you learned mathematics-based engineering approaches and chemistry-based engineering approaches and physics-based engineering approaches, and that's what you made your technologies out of. Biology had no seat at the table, okay, because it did not exist in any useful way at that time. So, in the biomedical engineering that has existed for a half century, it started out and has lived most of its life without biology. It might as well just be medical engineering, would really be the most accurate term. All right, so what's happened between then and now? Between the 1970s and 2005 and where we are now? Well, what happened was biology changed. All engineering disciplines are based on some fundamental science. We'll talk about that in a minute. But there could not have been an engineering based in biological science because the nature of it prior to these two revolutions just didn't allow any useful engineering to be done. So what were the two major revolutions? Well, one was the molecular biology revolution that most of you are too young to realize actually happened, right? It's just been there forever. That was really in the 1970s. We'll talk about that in a bit. And then the genomic revolution, that's more recent. Without those two revolutions in biological science, there would be no biological engineering. There could be no engineering rooted in biology without those two. So that was what was important. All right, so it really began with the molecular biology revolution and a couple of the key milestones. Cohen and Boyer with the first recombinant DNA, putting a new gene into bacteria, and then Kohler and Milstein on hybridomas to produce monoclonal antibodies. All these things are old hat to you now. Prior to these dates, did not exist. And this was crucial because engineers need components to manipulate and put together and behave the way you want them to. Prior to the molecular biology revolution, you couldn't do that. An engineer could look at any kind of tissue and say, well, that's kind of nice, but there's nothing I can do about it because I don't know what the underlying components are. So this was a huge deal. The problem was it was incomplete because it remained trial and error. You could go in and change genes however you wanted, but you couldn't predict what was going to happen because you could make that one gene change here or there, but we know now, of course, they knew then, that was in the midst of thousands of other genes and proteins, and so you couldn't predict actually what was going to happen when you made that change. You'd hope something good would happen. You'd hope something bad didn't happen, but it wasn't engineering because it was purely trial and error. So what really then nailed this down was then the genomic biology revolution that really started in the 90s, started with the Department of Defense, actually, and Charles Cellisi, NIH, Wellcome Trust, Whitehead, Sanger, onto the Broad, and this worldwide effort to first sequence the human genome, very much an industrial scale. Everything we talk about is omics ever since has depended on this, the ability to sequence in a high throughput way and find out all the components that were there. So the genomic biology revolution now allowed at least the notion of being able to be predictive about what would happen. Because even though you could modulate any one or two components, now if you could at least measure and understand the interactions with all the other hundreds or thousands of components, you actually had a chance of making predictive technologies. So one way to put it is there was this new fusion of biology and engineering. The molecular biology revolution permitted biology to be fused with engineering. Engineers could now do things in biology. I would assert that the genomic biology revolution required biology to be fused with engineering because you needed the ability to now comprehend complexity, quantify, computationally analyze, and so forth. So these were the two things that allowed a new biology-based engineering discipline to be imagined, where before both of these revolutions, it could not even be imagined. It was just really nonsense. All right, so we now had the opportunity to go from molecules and cells to systems, integrate multiple mechanisms, and affect higher level functions across the whole spectrum of applications. And so when we first started some of the BE curriculum, I later codified this in a little overview article, talked about the two things that really had to be learned by biological engineers was how to quantify the biochemistry and the biophysics going on, model it, predict it, manipulate it, and then also to comprehend the omic complexity so that whatever technologies you were coming up with, you had a chance of saying, yeah, what are they going to do in the system? And now the idea of applications, whether it was microbial ecology or energy or stem cells or antibodies and cancer, okay, that was up to you. One thing I want to point out here is this vision has always been educational in nature. The idea is if you're going to create a new discipline, it's because you want to teach next generations of people how to do things differently. So, some of you who are grad students here may recognize 2420 and 2440. These really are the cornerstones. That's why they're the requirements. At the undergraduate level, we kind of have these. We have also thermodynamics and transport and others, because at the undergraduate curriculum you can cover a wider range of things. All right, so this is what we got to. It became a new biology-based engineering discipline. And in selling this at MIT and many other places, I appealed often to historical analogies. Electrical engineering, mechanical engineering were physics-based engineering disciplines. Chemical engineering, materials engineering were chemistry-based. Both had analysis and synthesis, but those were their sciences. This was now a chance to do this with biology. And so to contrast it with the traditional medical engineering field, one thing I used a lot as an illustration, some people loved this, some people hated it, some people said, what the heck are you doing? But that was the point, that all engineering disciplines depend on some underlying science, and you do analysis and synthesis, and then you have applications. And all robust engineering disciplines look like this, that there's a very well-focused underlying science and a very wide range of applications. And that's why it makes a really good educational discipline. In contrast to other engineering fields that I would talk about, like medical engineering, there's really only one application. And you can aim at that application really just pure math-based engineering or physics-based or chemistry. You don't even need to involve biology if you don't want to. So that's why it's so different. All right, so that's the first third of the talk. I just wanted to say where we are, what this really meant conceptually and intellectually. And again, to many of you, this is old hat because this is your life. Okay, but I wanted to give you where it started from. In the second half, I'm going to give you a few minutes about my own journey, about, well, how did I come to thinking this way? And then the last third will be about, yeah, but how did this actually happen at MIT? And that's the hardest part. All right, so when I was in college, I hated biology because it was so phenomenological. Right, and many of you of my vintage will know that. It was very descriptive, very categorical, and as somebody who loved math and physics and chemistry, it was just hard to figure out how to apprehend biology. So it was the subject I had the least interest in. So I majored in chemical engineering, because if I liked math and physics and chemistry, well, that's what you do, and you put them all together. And that was my major, but I really found it unsatisfying. I had a number of summer jobs that I would work in the petrochemical industry on distillation columns and polymer reactors and so forth. And I said to myself, am I going to do this for 50 years? Okay? And great discipline, very valuable, just not satisfying for me. But, as I said before, this timing was just perfect because while I was in college and heading on to grad school, that's when the molecular biology revolution actually occurred. And I started reading those papers. I said, well, this is really interesting. This now has chemistry and physics in it. We can actually see what's going on a molecular level, and you can do things with it. Why don't we treat molecular biology the way chemical engineers treat chemical reactions? Okay, so that was starting to occur to me that, all right, it could become mechanistic, and that meant an engineer could do something with it. So I started dreaming something like this. This is one of my first complicated diagrams. And I said, well, what engineers do is they want to control and predict and produce the operation of a system. And they have all kinds of different components that have properties. And you put together a model and say, I need this component property and that one, and that's going to give me the system operation that I want. It's just that in biology, our components were these genes and proteins and so forth in all their activities and states. And the system operation was essentially cells. What do cells do? Because they're the basis of all biology, whether it's physiological, microbial, and so forth. So this became my dream. Why can't we do this in biology? Think like an engineer, but do it with biological components and biological systems. So my PhD, I was really fortunate. I had the two advisors at the University of Minnesota, Rutherford Aris, who was a terrific mathematician, and Kenneth Keller, who was a very traditional biomedical engineering. And I said to them, yeah, but I really want to do something with cells. And they said, oh. But they had a friend, squash partner, who was a microbiologist. He said, well, you know, there's this thing called chemotaxis. The cells wander around, but they can move directionally in gradients. We don't understand it. We don't understand how they do it. We don't understand how to quantify it. So that's what I did my thesis on, and it was essentially on microbial ecology, and looked at how these different properties of migration of cells would affect the growth of populations. Found this really neat engineering trade-off that too much of this random type motility was bad for population growth. More chemotaxis was good because they would go to the right directions. But to have chemotaxis, you had to have the random. So you had to have this trade-off of what was actually happening. So, okay, good. This means you can design bacteria to do what you want. Okay, so I had it. I said, this is my future. But there was no interest in either industry or academia for this kind of idea. So I really was at a crossroads. I had some offers, again, at some petrochemical companies, but I really wanted to do that. So what happened? Well, the world was a mess, but I got lucky. So the mess in the world, and again, this was before most of your time. How many of you remember this? Okay, the oil crisis in the late 1970s. Won't go into the reasons for it, but it was so, there was embargoes and so forth, and you had to get gas either on an even day or an odd day, depending on your license plate and things like that. So what that meant was the United States then said, wow, we got to pump a lot of oil, so we need a lot of chemical engineers. And so chemical engineers were getting paid a lot, so all kinds of high school students wanted to go to college to major in chemical engineering, and there weren't enough chemical engineering faculty members to teach them. So if you were breathing and got a PhD in chemical engineering, you could get a faculty job. No lie. They would call you up, say, hey, I hear you're going to finish in the next year or so. Yeah? Okay, would you like a job? But here's what was important about it. They didn't care what my research interest was. And I would say, yeah, but I really want to study cell biology from an engineering point of view. And so, yeah, that's okay. You know, can you teach transport phenomena? Yeah, okay, good. So that's the only reason I got an academic job, because otherwise nobody would have hired me for the research I was interested in. There was no industry that would have hired me. So it was only this that gave me a first step. All right, but we were able to get underway, and we were studying things like this. We wanted to study what governed how cells decided what to do. So we had the extracellular stimuli and intracellular networks and the responses. And this is just a beautiful engineering concept for quantitative modeling and measurement. And so this Q signals responses in fact, I found a cartoon, and I've kept it to this day. You probably can't read that, but this is a couch cell. I don't know if you're familiar with couch cells. And the couch cell spouse is saying, stimulus response, stimulus response. Don't you ever think? Because that was the approach we were taking to understanding what was going on in cell networks. Where we did it, I did get my first faculty position at University of Pennsylvania through the 80s, moved to University of Illinois in the early 90s for family reasons. And here's the important point that has to do with MIT and today in biological engineering. I was really unusual at that point in time. And I had appointments both in chemical engineering and in cell biology at these institutions. And in fact, at Illinois, and Dane would remember this, the cell biology department was very young, and I was one of the senior cell biology faculty members, led faculty searches and things like that. So what that meant was I really became familiar with the interface, the culture between engineering and cell biology, molecular biology. I was really living both, bilingual, if you want to call it that, and so forth. So it was good preparation for what ended up in the way biological engineering got created. But there was still a problem. This whole approach, this whole goal of engineering cells was not recognized as important by chemical engineering or bioengineering or cell biology. If I gave talks at either the AICHE for chemical engineering meetings or biomedical engineering societies, for over 10 years, my talks were always in the miscellaneous section. They would have a miscellaneous session at the very end. So you'd have a meeting that went till like Saturday afternoon, and the miscellaneous sections were always the latest one, and it was you and the other speakers, and you're kind of looking around. What are you going to talk about? I never heard of it. What are you going to talk about? I don't care. So that's what my presentations were. I did go to American Society of Cell Biology, but only poster presentations ever, because they couldn't see why any engineering approaches were of interest. This little book was profoundly annoying because it was put out by the National Research Council in the late 1980s, I think it was, and it said, these are the frontiers. If you're a chemical engineer, these are the exciting things you need to be working on. And so you get it, and you're a young faculty member, say, okay, good. Where's my work? This, and not a word. And if anything, it was caution against, well, don't be a second-rate biologist. That's not what engineers should be doing. So it was pretty discouraging. But that's okay. You will all face discouragement too, and you have your self-confidence and your energy, and you'll be fine. Okay, so what is it? Persisted, and an interesting thing was from the classes I'd been teaching, I put together a book, a monograph, with a really terrific early graduate student at Penn, Jennifer Lindemann, who had a great career at Michigan. Strange to say, she was one of my early students. She's retired now. And like I saw her to me, say, Jennifer, what's the deal? But everybody's different. So anyway, all of this thing about how you quantify what's going on in molecular interactions and signaling and adhesion and so forth, just really kind of all documented it here. So it had kinetics, mechanics, transport information processing. And the reason I'm mentioning this is where a lot of the ideas for a curriculum in biological engineering started to germinate. Because you say, okay, if you're really going to understand how to quantify and model and manipulate cells, you need to know all these different things. And there's a lot of different classes you would need to take in mechanics and kinetics and transport, information processing, and so forth, just addressing cells. Whoops. Interestingly enough, it wasn't until we moved to MIT that we started using it in a curriculum in bioengineering, as we started our curriculum here. But it actually, throughout the 90s and the 2000s, it was used in many biochemistry and biophysics and pharmacology programs. I would go visit people at other places and they say, oh, yeah, that's the book we use for our pharmacology course or our biochemistry course or whatever. Bioengineering? No. Until we started our program here. All right, so that's kind of how I got my own thinking. Whether that's important or not, interesting or not, it's part of the story. All right, so where does MIT come in? All right, well, these days, you got to look it up on your AI overview, and you say, what happened? Okay, well, timing-wise, it was 1995, and MIT did come to recruit me, because I did want to create something new. It didn't really have a bioengineering program. I had medical engineering in all the other departments, chemical and mechanical and electrical and so forth. But it didn't have its own department. So it wanted to do something, and because MIT was really the home, really the premier home of molecular biology and genomic biology, what better place to now try to bring to fruition this vision. So this gives you a few of the milestones. I'm going to talk more about it later. The important thing here was it was 1998 when we created this new academic entity. I'll talk more about it in a minute. But this is one reason I really wanted to show this slide. It says it was co-directors, Douglas Lauffenburger, okay, and Gerald Tannenbaum. Anybody know Gerald Tannenbaum? Okay, no, so don't believe everything that you get on your... I think that's what happened. Yes, I think that's what happened. It brought Jerry Wogan and Steve Tannenbaum together and said, well, it's got to be good enough. All right, but note to students, okay, careful. All right, so what had just been done here in the landscape in the early 90s? Well, a really nice undergraduate curriculum development was something called the biomedical engineering minor. Some colleagues you might recognize, Linda Griffith, Roger Kam, Al Grzinski, Paul Matsadera, Bill Dean as well, created a way to take students from any other major, chemical engineering, material science, mechanical engineering, so forth, including biology and chemistry, and teach them some extra courses that said how did their engineering interface with modern biology. Okay? But the crucial thing that MIT did right was say, yeah, but this is a minor. It's not a major for all the reasons I said before. You could major in any of these other things, add these extra courses, and say, okay, now I can apply my other major, and I can apply it to the medical field and problems and so forth. So this was on the books and was very popular, but it also was in the right place. It was a minor available to any other majors, because that's exactly what it was. All right, so how to create something new. It was interesting. When I got hired, the provost and the dean said, okay, we want you to create something new at the interface between engineering and biology. That's kind of open-ended. And the first order of business was how to convince the School of Engineering to do this. Because anytime you create a new academic entity, it takes resources. Where do resources come from? Either the president says, brand new resources, all yours, or it's taken away from other units. So in fact, at the University of Illinois, I had actually begged the engineering school to start a bioengineering department. And when I made the presentation to their engineering department heads, they turned to the dean and said, oh, will this take extra resources? Yeah. Where are you going to get them from? Well, I'm going to have to cut all of your budgets. So they said, nope, not happening. Okay, and fair enough. So how did we convince people to do that here? Well, Bob Brown had a brilliant idea, and he was the dean of engineering at that time. And he said, we've actually got to show the School of Engineering faculty that there is something important here. That if this is going to be the best engineering program in the world, we got to do something about biology. But most of the engineering faculty had no idea about these two revolutions. okay, because these people were all old enough that they had had the same biology I had had in high school. And so, well, biology, you can't do engineering with biology. So what he came up with was a summer short course in biology for engineering faculty. And Alan might recognize many colleagues here because it was taught by MIT biology faculty, and it had hands-on labs. So in the morning there were lectures, might be Bob Weinberg and Phil Sharp and Graham Walker and so forth. In the afternoon they actually did recombinant DNA and enzyme kinetics and things like that. And so you see a lot of here, some of the engineering faculty, maybe you recognize a few, maybe not. Tried to get as many department heads. And after this was over, then we could go to the engineering school and say, all right, what do you think? Isn't there a new science here? Isn't there the possibility of a new kind of engineering? And they said, yeah, I guess you're right. We had no idea biology actually was in this state now. This is actually pretty cool. So Bob really was brilliant in doing that. Okay, so the engineering school, okay, we can do something, but what do we do? Well, there was no administrative process in place to create a new academic unit because MIT just didn't do this. Okay, there's many kinds of institutions, especially medical schools, where you can create units easily. MIT very much centered on undergraduate education and disciplines. It just didn't do this very often, so it didn't really know how to do it. The provost then, Joel Moses, under the President Chuck Vest, just set an ad hoc committee to say, well, how would we create a new academic unit? Okay, ironically, it had nothing to do with bioengineering. There were people in systems engineering, some of my favorite colleagues that are still around, that said, we've got to have a new systems engineering unit. Okay, and so this special committee, ad hoc committee, to figure out, well, how do you create a new unit, was created to figure out what to do about systems engineering. Turned out they got created the same time we did. They are now IDSS. So it's kind of interesting to look back at that history. So it was chaired by Earl Merman of the Aeroastro department. And I was put on as a member by Bob Brown to say, hey, okay, if you figure out how to do this for systems engineering, maybe this is the way it'll work for bioengineering. Okay, what was created was this weird construct called a division, where faculty would have 1 foot in this division. I got it as BE here. At the time it was BEH, Bioengineering Environmental Health, and 1 foot in another department. And the reason for that was because these new units were experimental, maybe it would fail, maybe it would crash and burn, maybe it would be useless. And so it had to have the opportunity for faculty to just go back to regular departments if it failed and crashed and burned. So in 1998, there was a committee chaired by Bob Armstrong that said, okay, how would we do this for bioengineering? And yeah, we ought to be about 30 faculty total. That's about the size of most of the other engineering school departments. But what they're going to have is some will have 100% appointments, some will be 50% with another department, some be 0. Seems kind of arcane, but it was really trying to be protective about what would happen. Because here was the crucial thing. It will be reviewed after 5 to 7 years to evaluate. Continue indefinitely, become a department, go away. How would that be decided? And all kinds of criteria about educational programs, attracting faculty, and so forth. All right. So it started with 22 faculty, and some of the names you may remember, but you may know: Pete Didon, Bevin Endelward, John Essigman, Ramses Sekaran. See Jim Fox here. I think Steve Tannenbaum is on Zoom. A number of colleagues from School of Engineering departments, uniquely Paul Matsudaira from the biology department, and then some joint faculty, including Bob Langer and Harvey Lodish. So the faculty came either from other School of Engineering departments, by and large, and had this 1 foot in each camp. But some came from the toxicology division that Bevin mentioned, that had been at Whitaker College. What was Whitaker College? Well, it was largely faculty who had been in the Applied Biological Sciences department in the School of Science. And you'll see how those dots connect in a bit. All right, so there were some faculty from School of Engineering, some from the toxicology folks. And so when we started, Steve Tannenbaum and I were the co-directors to try to bring these two sets of faculty together, which was very interesting. Steve was a wonderful partner. We're going to have a nice celebration for him in a couple of weeks. I have to definitely shout out to Ronnie Dudley-Cowens. I'm glad Ronnie's here. She was the administrative officer from the start. Imagine she's been through all of this, through all these department heads, even beforehand. And she was very wise and thoughtful and smart. And boy, did she kept me out of trouble. Because in starting this up, we had very few resources of any kind. We were always just kind of making things up. Could we do this? Could we do that? Let's try that. And Ronnie would say, Doug. And I said, what, Ronnie? And we'd have to figure it out. So thank you for keeping me out of trouble. All right. I want to say a few words about culture. It's hard to encapsulate it. We really try to value people. Okay, we're as an educational institution, people are most important. Something I called out that I just really love in Essential Things Unity, you've got to have core values, standards, aspirations. But then liberty and a lot of other things where we can have a lot of diversity and heterogeneity and creativity and all things charity. Always view the folks that disagree with you with kindness, with generosity, respect, and so forth. So we've tried to institute this as the culture for students, faculty, staff from the very start. I have a couple things I want to show you. I used to show these in graduate student recruitment. Some of the faculty will remember these because I tried to explain to the students, prospective students, why would you come to MIT? MIT is such a nasty, cold, competitive place. They tried to explain that wasn't the case. So I said, all right, this is probably what you think MIT is like. How many people remember this? Okay. All right. So that was the idea. This is MIT. And we said, yes, okay, but that's not what biological engineering is going to be like. That is not going to be our culture. So what's going to be our culture? Collegial collaboration. What's a good illustration of that? Anybody remember this one? All right. So here's our antelope. Boy, does that need help from that cheetah? So if that's not collegial collaboration, I don't know what is. So graduate students seem to like it. They came. So. What I don't understand is why some of you have come and you didn't even get to see this at the admissions, right? Okay, so a couple years after we started, you started to have visiting committees. All departments have visiting committees to evaluate you, especially us, on our road to extinction or not, hence the dinosaur. So what did they say? Well, there's a sense of great pride in the department that it's doing important work, growth mode, opportunity. Big part was these weird appointments. Could they work? These shared appointments. Another big one was blending the bioengineering and toxicology faculty and cultures and education programs. And here I want to make a big point of recognizing bats. See, now you see where it comes from. Bioengineering and toxicology seminars, okay? And it had been a tradition already in toxicology, but this was a central vehicle for really bringing people together from what's research is going on in all these different labs. Oh, what are my thoughts about that might be relevant to Bevin's or Leona Sampson's or Jim Fox's? All right, so then we really got to be explicit. Okay, we really want to become the world-leading program fusing engineering with biology and actually define this new discipline. Historical analogy here to chemical engineering. Instead of chemistry-based engineering, biology-based engineering. Here was our first retreat. Brewster, Massachusetts. And for those of you who are grad students or postdocs now, you may be pretty envious. It was 2 overnights. We went for an evening, a whole full day, then a half day, and all came home. But it wasn't very many people. It was cheap, all of that. So, no, don't get your hopes up. But that's why I started. And it was really crucial because then we did spend a lot of time together. So who were the first faculty we added during that historical epoch? I told you I'm stopping at 2005, so Scott is the climax. First folks we hired, Dane Wittrup, Leona Sampson, Daryl Irvin, Bruce Teeter, Peter So. You can see their areas there that were very important for us: protein engineering, immune engineering, toxicogenomics, so forth. If they're in italics, it's because they were already on the faculty here in another department, and we brought over part of their appointment. There's Mike Yaffe, and if they're in gray, they've moved on, and that's okay. They provided tremendous value while they were here. But you can see a lot of names you recognize that became then the building blocks on top of the first set of faculty. A tough thing was we were spread all over campus, and we still are. Okay, but this was a difficult thing. We're trying to bring together all these disciplines to create a new one, and in a very collegial and interactive way, and yet we're all over campus. And tried as I did to get a new building and so forth, dedicated to our department, just didn't happen. So we had to figure out ways, when faculties in their labs were all over the place, to still create this integration and collegiality, and so that with things like the retreats and BATS and so forth. All right, so in 2003, only 5 years, there was a committee chaired by Raphael Bras of the Civil Engineering Department that evaluated us, and this was like, drum roll, exist or extinct. Foremost conclusion is that it's a success and has achieved its objectives, should be made a permanent academic unit and should become a department, should create a major, really emphasize a strong relationship with the Department of Biology, which was crucial. That was the science space we were claiming. We really wanted to be good partners with biology. So we were just thrilled. It's like Pinocchio. We're a real boy. So just to quick summarize the chronology, 1998, it was this division of bioengineering and environmental health on a trial basis. It was 22 faculty, started with the toxicology PhD program and these undergraduate minors. A year later, started the bioengineering PhD program. First visiting committee said we're on a good track. 2001, we said, okay, we're really aiming together for this new discipline. Let's dispense with bioengineering and environmental health. Just call it biological engineering. We were still a division. The new visiting committee said, this is going great. You actually should have an undergraduate major, because if you're going to be a discipline, you're going to teach undergraduates a new major. Oops, second one. We evolved now to have, then we had 2 tracks, had shared classes. 2003, as I said, we got approved as a permanent unit. Some of our space expansion was over into Tech Square, for better or worse. Gave us space, spread us out even more. 2004, I'll say more about this in a minute, started the new SISB program jointly with biology and ECS. 2004, the visiting committee said, yes, you should now have an undergraduate major. You should become a department. And then in 2004, all those things really got final institute approval. The new major, become a department, and gain a course number. I'll say something about that in a minute. And by now, we had moved from 22 to 38 faculty members. That's pretty good for 7 years, right? So this is what the retreat looked like then in spring of 2005. Only 1 overnight. And now, how many overnights do we do? 0. I just can't do it. It's expensive. But still, wonderful collegiality and just the best people. Just the best people. Just to point out the new major that did get started in 2005, you might, these numbers, BE 110, BE 320, because we hadn't gotten a course number yet, so there were still BE dots. But many of the classes here still exist. Some have been evolved. But now, this was the right kind of discipline. It was founded on the key aspects of biology, taught by our colleagues in the biology department, and then these engineering classes that showed you how to do analysis and synthesis, measure, model, manipulate, make, on these biological components. And then finally in 2005, and this is kind of where the story ends, we got a course number. Now, to some people who are grad students or postdocs from other universities say, what's the big deal about a course number? It just makes things more confusing. At MIT, it's the coin of the realm. If you've got a course number, yes, it's Pinocchio. You're real. But we could not have a course number until we became a department and had an undergraduate major and so forth. So interesting, we picked course 20. We could have picked any number that we wanted, honestly. Well, I don't know if they would have allowed us to have 73 or something, but we picked 20. Why? Well, there's history here, too. Course 20 had been established in the 40s, initially as the Department of Food Technology in the School of Science. It was in the School of Science. And it evolved a lot, nutrition, food sciences, nutrition and food science. In the 80s, became applied biological sciences. And then in 1989, that department was disbanded by the dean of the School of Science. And I'm not going to go into that. I wasn't even here. So I just don't ask me. But there were reasons. So it was disbanded. And so since 1989, that's why the faculty from the Department of Applied Biological Sciences moved into the Whitaker College to be this thing of the Division of Toxicology. Some went into other departments, chemical engineering, chemistry, and so forth. But that's really where a lot of biotechnology started. And so when we got to pick our new number, it just made a lot of sense that what had been applied biology in the School of Science, well, most applied things are in a School of Engineering, so it made all the sense of the world that biological engineering was essentially now the modern day applications of biology to technology and engineering. So we took on course 20 and gathered in all the alumni from the previous decades. All right, so since then, and I just wanted to point out, again, it's research is spectacular across the entire campus, including our department. But one thing that we've really tried to do was have impact in education. So I just want to list a few education-related innovations that were either led or co-led by BE along with other departments. The first thing that I'm still just very proud of was creating computational systems biology with biology and ECS. It still lives on in this CISB graduate program and in the Biomicro Center. That's crucial to campus and just helps everybody. And so it's a wonderful partnership back then with biology and ECS to do something like this. Then the microbiology graduate program with biology, chemical engineering, civil engineering. Similarly, iGEM. People have heard of iGEM? Okay. That actually spun out of our labs. Bevin, Natalie, right? Okay. BioBuilder is now a educational program in graduate schools, elementary schools and grade schools, very influential in getting kids interested in biology and biotechnology. The COM Lab started in BE, Eric Alm's idea. For graduate students, the BE Application Assistance Program, BEAP, that was an innovation brought by the grad students. NEED program, Living Machines, Biomakers with chemical engineering. This kind of goes on, and I'm just really happy about this because it is impact on our campus and beyond. So to finish, I'm just going to go back and say, I think it's true that BE is living impactfully ever after. And I don't know if you can read that. We had T-shirts and mugs and hats, and we had this is the mooing turtle. Okay, and we also had what else did we have? The what? The oinking whale. Yep. The barking cow. Right. So because it's just showing you can do engineering and biology. If that doesn't grab you, that's okay. There's many things we can do. All right. Thank you for listening. I hope this was of some interest.